Welcome, guys. This is part 2 of the Chapter 43 lecture for AP Biology. I left off talking about some of this stuff, but I was stumbling over it, so I thought I should take a break and come back and get back at antigen receptors of B cells. So notice we're binding to specific antigens. We've got those heavy chains and those light chains. So the tips of the chains form that constant region, and then the variable region. That's going to allow us to be much more specific. So think about the adaptive value of variable regions, and that essentially the adaptive value would be that secreted antibodies are structurally similar to those B cell receptors, but they lack those transmembrane regions that anchor the receptors to the plasma membrane. So they're essentially receptors that don't have a cell behind them. If that makes sense. So T cell receptors, 2 different polypeptide chains. Again, they're going to be tips that are V and C, but they're straight line receptors, so they're going to be a little bit different in terms of which they can bind with, if that makes sense. So T cells are going to bind to antigen fragments. Those fragments are bound to cell surface proteins called MHC molecules. That should sound familiar. Major Histocompatibility Complex. That's going to be how we're going to recognize our self cells, if that makes sense. So yeah, that's definitely MHC and T cells are definitely A-level items. So MHC, they're going to bind and transport antigen fragments to the cell surface. So assuming that this is some sort of pathogen that has infected a cell. Excuse me. So one of my cells can recognize that they've been infected. It's going to then express these antigen. A nearby T cell recognizes, hey, something is wrong there. Something needs to happen. Depending on which antigen you are exposed to, you're going to have a different type of MHC molecules. So here's kind of what you're talking about there. Here's a model and a more realistic model, a more realistic example. Looks like a hot dog in a bun, right? That's kind of the idea. Excuse me. So class 1 MHC molecules are found on almost all nucleated cells of the body. That's, like I said, that's how we recognize ourselves. They're going to use class 1 MHC to display peptide antigens to cytotoxic T cells. You definitely need to know what cytotoxic T cells are. Here's a picture of that. So here's our cytotoxic T cell. Recognizes the class 1 MHC that's being presented. Excuse me. So this cell's been infected. That's going to, in the cytoplasm, the antigen fragment, so the piece of the pathogen, is going to activate this class 1 MHC molecule that's then going to be expressed on the membrane of the cell. When that's expressed, a cytotoxic T cell says, hey, I know you, and I'm going to do something about it. Over here, we've got a class 2 MHC molecule, and we've got a helper T cell that's going to say, hey, I know you, I'm going to do something different with you. Class 2, like I said, are going to be located mainly on dendritic cells, but they're B cells, antigen-presenting cells that display antigens to cytotoxic T cells and helper T cells. The helper T cells are going to help T cells. So lymphocyte development, we've got 3 really important properties that we need to kind of nail down on lymphocyte development. So how can we be so specific and so diverse? Well, we've got those conserved regions and those variable regions. That allows us to just manipulate the variable region. We're essentially just manipulating the receptors. We don't have to create an entirely novel molecule. We just have to create a novel receptor. We're going to use the same conserved regions. We're going to use the same light and heavy chains. We're just going to use different variable regions. Immunoglobulin gene encodes one chain of the B cell receptor. Many different chains can be produced from the same immunoglobulin gene by rearranging the DNA. We'll talk lots about that in the spring. I'll tie you back to this stuff. But essentially, we can move our genome around. We can express different proteins differently, and that's going to allow us to have different receptors on our B and our T cells. So here's a stretch of DNA, undifferentiated B cell, so generic B cell. DNA is going to be randomly selected between V and J segments, and then we're going to cram some stuff together. Then we're going to process that DNA. We're going to we're going to transcribe it to RNA. Then we're going to process the mRNA transcript. And then we're going to take it out and we're going to translate it, and that's going to give us our specific B cell receptor. Notice that the V and the C constant region doesn't change. The variable region does change. So depending on which V we choose, we're going to have a different receptor. The constant region is still going to produce the constant region. So essentially, I've kind of been talking through this, but you should be asking yourself, how do we keep from having autoimmune disorders on everything? Well, antigen receptors are generated by random rearrangement of DNA. So we're randomly making these antigen receptors. As the lymphocytes mature, they are tested for self-reactivity. If they recognize that, if they hit on our own cells, we just destroy them immediately through apoptosis, or we just turn them off. We can do that as well and just keep the packaging. If they weren't destroyed, we would all essentially have autoimmune disorders all the time. Okay, moving forward, we're looking at how do we amplify lymphocytes, or how do we make more lymphocytes. We're not talking about necessarily expression, but how do we take a lymphocyte that exists and make many copies? Because we don't need one copy, we need many copies. Essentially, it's going to use the process of mitosis. We're going to use clonal selection. So we're going to produce two types of clones of the mature lymphocyte. So let's move on to the next slide. Sorry, I messed up my slides here. So notice that there are two types of cells, effector cells and memory cells. The short-lived effector cells are going to cause the effect. They're going to fix the problem. They're going to destroy the invader. Memory cells are going to be around for a long time, and they're going to accelerate our response to our next exposure. First exposure to a specific antigen represents our first response, or our primary response. We're going to create effector B cells. They're called plasma cells. And we're going to create T cells. They're going to be activated to their effector form. So we're going to have effector B and effector T's going to do some work. In the secondary immune response, that happens faster because we've already been exposed to the antigen, so we've already got those B cells and those T cells. If we look here at our graph, now there's a lot going on here. Focus on the A line. Okay, so we've got a primary immune response to A. Excuse me, we're initially exposed to it. It takes a few days to build up, almost a week to build up antibodies. Then when we're exposed to A and B, quickly, our lag time is much smaller. Quickly, we get a whole bunch of antibodies, and we get more antibodies in our second exposure than we do our first exposure. Then at day 28, we were exposed to B, whatever antigen B is, and we get our primary response there. So I think this is just here to show the difference in the scale between the two. It's a much stronger response the second time. Think about your flu shot. Your flu shot was your first exposure. You developed a few antigens, and then next time you encounter the flu virus, you're going to have a whole bunch of antibodies in a hurry. I think I said antigen there, and I meant antibodies. You know what I mean. So acquired immunity defends against infection of body cells and fluids. That's called our humoral response. Sorry about that. Which is the activation and clonal selection of B cells. And then we get our cell-mediated immune response. It's going to involve those cytotoxic T cells. Helper T cells are going to help in both. So, of course, here's a picture for me to talk from. I know this is really small, but left side there you're seeing the antibody-mediated or the humoral immune response, and the right side you're seeing the cell-mediated immune response. So notice that we get our first exposure here. So let me widen this a bit so we can see a little bit better. Okay, so we get our first exposure. That's going to immediately trigger B cells to be created. It's going to create those plasma cells that are going to be the effector cells, and it's going to create some memory cells. At the same time, the antigen is going to be engulfed by an antigen-presenting cell that's going to jumpstart the cytotoxic T cells. Helper T cells are going to be involved, but that's going to basically jumpstart the secondary response. So we've got two defense mechanisms happening at the same time. Zoomed in version, moving on. So helper T cells are going to respond to almost all antigens. There's a special surface protein called CD4 that binds class II MHC molecules. This binding is going to help keep the helper cell attached to that cell while activation occurs. It's sort of like when you had to go get help, but so like say for example, I come across someone that needs CPR. I'm not just going to run to go see a doctor. I'm going to stay with that person and help that person until someone more qualified can show up and really help them. Same thing is happening here. The helper T cells are going to stay with that antigen-presenting cell until someone bigger and badder can come and can destroy that cell. There's kind of what you're seeing in that picture. So let's look at cytotoxic T cells in particular here. Notice they're going to make CD8. So these CD4 and the CD8s, I want you to kind of file those away in the back of your memory. When we start looking at HIV and how HIV infects cells, those same receptors are going to come up. So CD8 is a surface protein that greatly enhances the interaction between a target cell and a cytotoxic T cell. Think a normal handshake versus how President Trump shakes hands. Or maybe even better would be how they shake hands in Game of Thrones. If you don't watch Game of Thrones, the best way I can say it is like if I'm shaking your hand, my hand goes almost to your elbow and your hand goes almost to my elbow. So it's a really strong interaction. It really makes that bond really strong. Binding to a class I MHC on an infected cell activates that cytotoxic T cell and makes it an active killer cell. So it turns it on or it's off until it binds to MHC, class I MHC on the infected cell. The activated cytotoxic T is going to secrete proteins that are going to destroy the infected target cell. So here's a picture. We've got our cytotoxic T cell on the top there. We've got our class I MHC, and our CDH is really strengthening that connection. Our cytotoxic T cell, okay, so perforin is going to allow it's going to perforate the membrane, so it's going to allow things to be released. It's going to put holes in the cell membrane. So we're going to secrete these chemicals. They're going to be taken in by the target cell. The target cell then is going to shrivel. As it shrivels, it's going to release the cytotoxic T cell, and the T cell is going to be inactivated. That, I think, is a really good stopping point for video 2. I'll pick up with B cells in lecture 3.